Hello, my name is Carol Leibel, and today we're going to examine how eukaryotic cells control the expression of genes and the production of proteins. And the regulation of these genes is different from prokaryotic cells, and there are several reasons for that. In eukaryotic cells, they have a lot more genes than prokaryotic cells. For example, humans have 23,700 genes in their genome, compared to a prokaryotic cell like a mere E. coli that averages 3,000 genes. In addition, there's a lot more DNA in a eukaryotic nucleus. In humans, there's over 6 feet of DNA packaged inside the nucleus. In order to manage all that DNA, it's wrapped around organizing proteins called histones. And in addition, there are more non-histone proteins involved in gene regulation than you would find in a prokaryotic cell. Now, here's the crazy part about eukaryotic cells and multicellular organisms. Almost all the cells in an organism are genetically identical to one another or totipotent. The term totipotent means total, um, total potential. And so theoretically, I could take one of your skin cells and I could clone you. Now, you may not like that, but I could do that. So if your skin cell, your muscle cell, and a fat cell has all the same DNA, why are they so different from one another? The answer to that is that there's differential gene expression, and that means that in these cells, different genes are activated and deactivated from one another, and that will produce different kinds of proteins, which will give rise to different types of cells. While genes are being transcribed, there can be mistakes in this regulation, and mistakes in gene regulation can give rise to diseases, including cancer. So it shouldn't surprise you that gene expression is regulated at many stages and in many different ways. The first thing we want to look at is the packaging of the DNA, because the packaging of the DNA can also impact which genes are going to be expressed. So we know that the DNA is a double helix and that there's a lot of DNA in the nucleus if you're talking about a eukaryotic cell. Here we're looking at the DNA being wrapped around those organizing proteins called histones, and in this case there are 8 of those histones, giving rise to a structure called the nucleosome. So here is a micrograph showing these nucleosomes attached to a strand of DNA. It kind of looks like a strand of pearls. These nucleosomes then are going to be packaged rather tightly to give rise to chromatin fiber. And that chromatin fiber is going to be looped and attached to a scaffold protein. And then that protein is going to fold on itself to give rise to a chromosome that we can see during the process of mitosis underneath the microscope. So we want to look at the DNA being packaged in a little video.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Well, it turns out that less than 10% of our DNA is actually expressed into protein, and probably closer to 1%. What happens to the rest of that DNA, you might be asking? When you studied protein synthesis, you remember that there were nucleotide sequences within a gene that were never expressed. We called those intervening sequences, or introns. An average human gene is 36,000 base pairs long and contains 6 introns. There are about 5,700 base pairs each. In addition to that, there are 7 exons on the average. However, they are only 300 base pairs each. So the actual coding region is only 2,100 base pairs long for a gene that's over 36,000 base pairs. So that implies that a lot of that gene is never used. In addition to that, in between the genes, we have found that there's what we call repetitive DNA, and there's 2 types. One is tandemly repetitive DNA, and so here is an example of that: GTA, 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 GTA. All right, and so this is the repeat, and because these repeats sit side by side, they're called tandemly repetitive DNA. Now, we can find tandemly repeated DNA in between genes, but we also can find it at the end of the chromosomes. And the tandemly repetitive DNA at the end of the chromosomes, they're called telomeres, and they protect the chromosomes because as the DNA is replicated, the lagging side of the DNA, you will recall, becomes shorter. And so it protects the DNA as it's being lost from actually doing any kind of damage to important genes. Now, in addition to that, we also know that there are larger repetitive pieces of DNA. However, those larger sequences or larger segments um, are not found necessarily next to one another, and they're repeated throughout the DNA genome. Over time, many of these larger sequences experiences mutations, so there may be slight differences between this interspersed repetitive DNA. It's estimated that 25 to 40% of all the DNA for mammals is this interspersed repetitive DNA. Now, you've got me because in talking about gene regulation, we've got to talk about a few exceptions to the rule. I know that I just said that the most of the cells in a multicellular organisms that it's totipotent, but we want to talk about a few exceptions. One is called gene amplification, and that means that you have a particular gene that is duplicated, maybe one time and maybe many times. An example of gene amplification has been found in the amphibian ovum or egg, where inside that egg there's over 1,000,000 copies of the ribosomal RNA, which are made from tiny circles of DNA that has been duplicated from the genome. On the other hand, something that's just the opposite is called gene loss. An example of gene loss occurs in gall midges, and this is an interesting phenomenon because during development, in all but 2 cells, 32 of their chromosomes are lost. Now, where they go, I can't tell you, but they're lost. So the majority of their cells only have 8 chromosomes, but the 2 cells that have 40 chromosomes are going to produce the gametes so that the offspring start off with 40 chromosomes. Another example has to do with transposed genes. It's not unusual for a particular gene to be duplicated and moved from point A to point B. over time, that duplicated gene will experience different kinds of mutations than the original gene, which may give rise to different kinds of proteins. This may give rise to something called a gene family. And the gene family that probably most people are familiar with is the hemoglobin gene family. So this is a diagram of the hemoglobin gene family. And so think of this as evolutionary time. And here's the ancestral globin gene. Now, the hemoglobin that you have in your body has 4 polypeptide chains. 2 of them is called beta hemoglobin and 2 are called alpha hemoglobin. So at some point in time, there was a duplication of the ancestral gene, and it actually was transported from one chromosome, chromosome 16, to chromosome 11. And that gave rise to the alpha and the beta forms. And so once they are on those chromosomes, they themselves were duplicated and experienced different kinds of mutations, so that today, if you look on chromosome 16, there are actually 1, 2, 3, 4, 5, 6, 7 copies of the alpha gene. All right, and over here on chromosome 11, there's 1, 2, 3, 4, 5, 6 copies of the beta gene. Now, what's interesting are these little green genes, if you will, and those are called pseudogenes. Those are genes that were duplicated and they experience mutations. However, those mutations then gave rise to a non-functional gene. It's not transcribed or translated, and so they're called pseudogenes. You'll also notice that there are some genes that give rise to different kinds of hemoglobin. For instance, the hemoglobin you had as a fetus is different from the hemoglobin you had today. And that's because that fetal hemoglobin has to be able to have a stronger affinity for the oxygen to be able to obtain it from the maternal hemoglobin. At any rate, so this is an example of gene duplication that gives rise to gene families. All right, so I want to talk about another concept, and that is sometimes when a gene may experience gene rearrangement. It doesn't happen very often, but the example that I'd like to go over happens with your B cells, which are part of your immune system. Now, B cells give rise to all kinds of different kinds of antibodies that will fight disease. In fact, it's estimated that they produce millions of different kinds of antibodies. Now, seeing that you only have 23,700 genes, the question is, how could that be? And that the reason is because the genes that give rise to the antibodies have the ability to rearrange. So let's look at this little diagram. Okay, here are the genes that give rise to antibodies. Now, once this cell comes into existence, you can see that there's a deletion of this particular part of the gene. Okay, so now we have, and these are called the variable regions, so now we have that exon and that exon. When it comes time to transcription, it turns out that only this particular intron is going to be transcribed with the rest of the, not introns, but exons, with the rest of the exons. And now what happens is that this is going to give rise to that part of the antibody that's going to be unique for that particular lymphocyte. All the lymphocytes experience different rearrangements of these genes. So now they have the ability to give rise to different kinds of antibodies to fight disease. We have a little video here that talks about another example of gene rearrangement in mimicry in South America.
At first glance, it seems simple to tell one type of butterfly from the next. Their boldly coloured wings appear to differentiate one species from another. But predators must beware. The wings of butterflies tell lies. Some butterflies have evolved wings that resemble those of more poisonous species, a clever deception. In other cases, two equally poisonous species have evolved together to become mirror images of each other. The Amazonian butterfly Heliconius numata has evolved in this way. This one species has many different wing patterns copied from several different species from the Melanea genus. This kind of shared evolution is called Müllerian mimicry. Together, Heliconius and Melanea spread an important message to would-be predators. Don't eat us, we taste bad. Their similar appearance makes them part of a cooperative that protects them from being eaten. While scientists knew Müllerian mimicry was at work in these butterflies, it was unclear how Heliconius could derive so many different wing patterns from only one set of genes. In the mid-1970s, scientists proposed that the wing mimicry of Heliconius was under the control of something called a supergene, a long stretch of DNA that contains many smaller genes. In the supergene, individual genes are immune to the random mixing and matching that normally occurs in sexual recombination. This is how Heliconius manages to keep its various wing patterns separate. Although the concept of a supergene fits with what scientists observed in the field, no one has ever seen how it works, until now. A study of Heliconius DNA led by French scientist Mathieu Joron shows that the supergene breaks and then recombines into several different configurations. Each arrangement creates a specific wing pattern, the mirror image of a Melanea species. By mimicking its poisonous cousins, Heliconius is safer from the greedy gaze of predators, so natural selection favours these preset patterns, and by extension, the genes that create them.
All right, the next concept we'd like to look at is called epigenetics, and it refers to a process that influences gene expression without changing the underlying DNA. There are two examples of epigenetics, and one is acetylation and the other one is methylation. In acetylation, you have your histone histones, and they have little tails, and they have lysine in them, and they can attract acetyl groups. And when the acetyl groups attach to these tails, it kind of causes the tails kind of cause the proteins to get larger and basically unpackage, you will, the nucleosome. And so now the DNA is exposed and is more likely to be transcribed. Okay. Now, methylation is just the opposite of acetylation. It tends to repress gene transcription. So a methyl group can be added to the nitrogenous bases of cytosine that's followed by guanine. And many human genes have what they call an upstream cytosine guanine rich region called CPG islands. The methylation of a gene's CPG island represses gene expression. Different genes have different methylation patterns which contribute to differences in gene expression in different gene types. So if you have a cell that has a certain gene that has been methylated, when that gene undergoes mitosis, those daughter cells will retain the methylation of those genes and they will never be transcribed. Another interesting phenomenon has to do with females and the second X chromosome. The second X chromosome in females is never transcribed or used, and we know that underneath the microscope we can see that second chromosome being bunched up and folded up, forming something called a bar body. Upon closer examination, it's been found out that the second X chromosome is highly methylated. And so that's the reason that none of those genes are transcribed, is because they're highly methylated. All right, so there are transcription factors that are also involved in whether a gene is going to be transcribed or not. As we mentioned in prokaryotic cells, there are regions right prior to a gene that's called a promoter region. So this right here represents a promoter region, and that's where the RNA polymerase is going to attach. They have found that in many genes, a little distance away from the promoter region, there are these proximal control elements. And proximal control elements will attract certain transcription factors that will help facilitate the attachment of the RNA polymerase. In addition to that, some distance away upstream, there are regions of the DNA that will attract enhancers, and these enhancers will attach to the DNA. They will be attracted to this region, to the other transcription factors, and the DNA is going to fold on itself. And when it folds on itself, what will happen is that will help facilitate the attachment of the RNA polymerase to allow transcription to take place. Okay, so here's the promoter region. Oops, sorry, let's go back. So here's the promoter region, all right? And within that promoter region, we're going to find the attraction of transcription factors, all right? And here will be the proximal control region. And then upstream, this is the distal control region. There will be the attraction of the activators. The DNA is going to fold on itself, and this will all help facilitate the attachment of the transcription factors and the RNA polymerase, and now transcription is going to take place. So we have a little video that will illustrate this process.
In eukaryotes, DNA transcription begins when general transcription factors bind to the promoter region of the gene. Those transcription factors facilitate the binding of the RNA polymerase enzyme to the start position of the gene. RNA polymerase needs to be activated by a type of transcription factor called activator. Some activators recognize and bind to the regulatory region of the DNA near the promoter region. Other activators bind to a distant region of the chromosome called the enhancer region. Activators bound to the enhancer region work with mediator molecules to initiate transcription. Another type of transcription factor, called repressor, inhibits the activation of transcription. Repressors can act in different ways. Some repressors bind to the binding site of activators and prevent activators from binding to DNA. Some interfere with the molecular interaction between the activators and the RNA polymerase. Some activators and repressors act by affecting chromatin structure. These repressors can alter the chromatin structure to make it coil up tightly. Coiled up genes are not available for transcription. Conversely, activators can uncoil a coiled section of DNA and make genes in those regions available for transcription. The interactions of all these factors determine the actual rate of DNA transcription.
So this diagram illustrates that different cells are going to have different activators. So if you're talking about a liver cell is going to make an albumin protein. A lens cell is going to make a crystalline protein. So certain activators are needed. And so looking at this, it's the combination of different kinds of activators that will allow the genes to be produced. You might think that there are thousands of different kinds of activators and transcription factors. However, that number is not as much as you might think. It's actually the unique combination of these transcription factors and activators and enhancers that allow a particular gene to be transcribed. Now, in addition to that, certain inhibitors are going to be needed to make sure that, for instance, this crystalline gene is not transcribed in a liver cell and that this albumin gene is not transcribed in the lens cell. So again, each cell has a unique combination of activators and transcription factors that will only activate certain genes needed for that cell. So I want to tell you the interesting story about being lactose intolerant. In order to digest a sugar found in milk called lactose, you need an enzyme called lactase. Now, when you're young, most people have the lactose gene and it produces lactase. As you grow older, most people do not drink milk or dairy products. So it doesn't make sense to continue making lactase. So there's a regulatory protein that is produced that will cause the lactose gene to inhibit it from being transcribed. Now that gene, that regulatory gene, is called the MC-M6 gene. And so as you age, this gene becomes activated and it prevents the lactose gene from being transcribed. However, there are people in this world in which there's a mutation in that gene. And if you have a mutation in that gene, then you will continue to produce lactase. And as an adult, you can drink all the milk you want and eat all the ice cream and have all the cheese. It's wonderful. All right? But it's because there's a mutation in that regulatory gene. And so they've kind of changed the naming of this phenomenon. It used to be people who couldn't drink milk when you were older, they were called lactose intolerant, and it was thought that they had some sort of mutation. Well, it turns out they're the normal ones. The folks that have the mutation are the ones that can continue to drink milk and dairy products. And so they now have a new naming system. The people who have that mutation and that regulatory gene, they're said to be lactose persistent. And the people who can't drink lactose as adults, they are the normal ones. So that's kind of interesting. Eukaryotes are a little bit different in a lot of their biochemical pathways, in that a lot of genes needed for, or a lot of proteins needed for a particular biochemical pathway, the genes are not located close to one another. And quite often they're actually found on different chromosomes. So the question is, how can you activate genes needed for a common biochemical pathway? Well, an example of that happens with testosterone. So let's examine what happens with this particular pathway. So testosterone is a hormone. It's a steroid hormone. It has the ability, remember, to actually come into the cell. Once it comes into the cell, it's going to attach to a receptor protein. Now it forms a hormone receptor complex. When it goes in, it activates more than just one gene. It activates a number of genes. So a number of different mRNAs are being produced. And so now all of them will be translated and can now participate in a common biochemical pathway. And so the coordination of the expression of related genes in a particular biochemical pathway may be facilitated by actually one particular hormone. So we want to look at post-transcriptional control, which is done by alternative splicing. Here's an example of alternative splicing that produces two different kinds of protein product, and it happens in Drosophila. Here is a particular gene, and it's called the double sex gene, which I think that's just adorable, but it's called the double sex gene because if it's processed in this way, in which we only have exon 3 and 4, it's going to produce a particular hormone that's going to give rise to a female. But on the other hand, if this gene is processed so that it has exon 1, 2, 3, 4 is missing, 5, and 6, it's going to produce a hormone that's going to turn around and give rise to a male. Remember, it's the spliceosome that's going to come in and perform the removal of the introns and also responsible for the alternative splicing. And so here's another interesting example that just happens in Drosophila. This is a gene called the tropin T gene, and this is most phenomenal because there are enough exons and introns in this particular gene that the messenger RNA can give rise to over 17,000 different kinds of messenger RNAs. And of course, that's going to give rise to 17,000 different kinds of proteins. Well, what in the world are these proteins going to do? These proteins are unique proteins, and they're going to be found on different kinds of nerve cells in Drosophila. You might be asking, well, does this ever happen in humans? Well, the answer is yes. You can't imagine how surprised the scientists were when they sequenced the human genome. They were really expecting over 100,000 different kinds of genes. They have found out that there are only 23,700 genes. But yet we can produce over 160,000 different proteins. How can that be? This is possible due to, again, alternative splicing during RNA processing. It's estimated that for each gene, there can be, on the average, 17 different proteins, and that 95% of all our human genes undergo alternative splicing. Here is an example. This right here is a DNA gene. However, in the hypothalamus, the gene is spliced in this manner so that it gives rise to something called the calcitonin-related polypeptide. It's a hormone that is only 37 amino acids long, produced by the hypothalamus found in your brain, and this molecule interacts with receptors in nervous tissue. And it's a potent polypeptide vasodilator that can function in the transmission of pain and is thought to be a cause of migraine headaches. On the other hand, this particular gene in the thyroid gives rise to calcitonin, and calcitonin is responsible for increasing the concentration of calcium in your blood. All right, so now we have our messenger RNA. How long does a messenger RNA last? A messenger RNA doesn't have an infinite lifespan. In fact, a lot of messenger RNAs are actually destroyed. And there's a way that the messenger RNAs are regulated that's recently been discovered. And it has to do with what they call RNA interference. And so I'm going to go through this and try to explain it. And we have a video to try to explain it because it's a little complicated process. So we have found that there are regions in the DNA in which it produces RNA. Now, once the RNA is produced, it's not messenger RNA, but this RNA that is produced has some complementary sequences. So what will happen is that it will turn around and it will bind with itself, kind of almost like double-stranded DNA. So once this RNA makes this like hairpin loop, it's going to go out into the nucleus, whereupon a protein called dicer is going to cut the end of the DNA, the RNA, that's double-stranded. And when it cuts that, the RNA now becomes double-stranded, and now it's going to be cut into smaller sequences. Once it's cut into smaller sequences, it combines with a protein called agarnat. Once it combines with the protein agarnat, what happens is that one side of the RNA is going to be released so that it becomes single-stranded. So now it becomes single-stranded. The agarnat with that little sequence of RNA goes around and it starts kind of like frisking messenger RNA. And if it finds some messenger RNA it can bind with, what happens is that it will cause the ribosome to fall off. It'll halt translation. And now what's going to happen is that messenger RNA is going to be destroyed, if you will. And so this is called interference RNA. All right, so here's another diagram of it. Like I said, there are two examples of interference RNA. One is called microRNA. MicroRNA, the origin of the RNA actually comes from a region of the DNA. So here is that RNA. It's going to, as I said, bind with itself. It's going to leave the nucleus. Here is that dicer that's going to basically cleave that into smaller pieces. It's going to attach to that agarnat. When it attaches to the agarnat protein, it's now going to become single-stranded, and now it's looking for messenger RNA that it can bind with, and when it does, that messenger RNA is going to be destroyed. Now, sometimes the origin of this RNA right here is not always from the DNA. It can come from a double-stranded piece of RNA. It can come from out in the environment, or now they've actually been able to inject that double-stranded RNA. That RNA that comes in that manner is not called microRNA. It's called silencer RNA or siRNA, and it's a little bit longer than the microRNA. So we have a video to look at, and it will explain it. I hope it does a little better job than I did, but let's see, okay?
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
All right, so there are some other factors that can affect gene regulation. For example, it's known that about 80% or 50% of all the messenger RNA never reaches the cytoplasm. So that implies there has to be some sort of chemical attaching to the messenger RNA that will allow it to leave the nucleus. Once the messenger RNA is in the cytoplasm, it appears that its lifespan is also determined by the length of the poly-A tail. Every time the messenger RNA is translated, it appears that the poly-A tail is shortened, and so that the length of the tail seems to indicate how long the messenger RNA will be around. So messenger RNA can last from minutes to just weeks in the cytoplasm. Once a protein is made, a protein, just like the messenger RNA, doesn't last forever. So how long or what causes a protein to last? Well, one of the things that appears to be a factor is the addition of a tag, if you will, to the protein. And in this instance, that tag is called ubiquitin. So when this tag ubiquitin is attached to a protein, that tags that protein for destruction. There is a structure called a proteasome that takes the tagged protein and breaks it down into polypeptide fragments and amino acids so that they can be recycled. And then the ubiquitin is put back together again, and then it's going to be recycled and the process starts all over again. So we do have a little video that explains this business of ubiquitination, if you will.
Often, polypeptides must be modified in some way to produce functional proteins. For example, a polypeptide is cleaved in two. Other molecules might be attached to the protein, or the protein might be phosphorylated. Some proteins need to be transported to specific destinations. A cell can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tagged protein.
So in addition to proteins being destroyed, in order for certain proteins to be functional, they have to have an addition of perhaps prosthetic groups. So for instance, hemoglobin, in order to be functional, has to have the addition of iron in a heme group. So let's suppose you're a person who doesn't have enough iron. If you don't have enough iron, then your hemoglobin will not be functional. In addition to that, sometimes the proteins have to be modified. For example, every protein theoretically starts off with the amino acid methionine. But if you look at these proteins, the majority of proteins do not have that methionine amino acid. At some point in time during a biochemical pathway, that particular amino acid or number of amino acids are cleaved. And so it's not unusual, perhaps through the Golgi apparatus, that proteins are modified, things are added to them, parts are removed, parts are rejoined in order to make a protein functional. So there is a post-translational control that has to take place. All right, so here we are. We're at the very end. And so this diagram gives you a nice summary of the eukaryotic control in regulating regulating genes in the production of proteins. So examine it, look it over. I hope this PowerPoint helped you understand the complicated process of gene regulation in eukaryotic cells. Have a great day. Hope to see you again. Talk to you later. Take care. Bye.